Living organisms have diverse forms, but their chemical makeup shows similarities. The difference between living and non-living matter is the amount of each element present. Carbon, hydrogen, and oxygen are found in different proportions in living organisms compared to the Earth's crust. Oxygen makes up 47% of the crust, but 65% of the human body. Hydrogen is 0.14% of the crust, but 10% of the human body. Carbon is 0.03% of the crust, but 18.5% of the human body. Scientists break down living tissue using trichloroacetic acid to analyze its molecular structure. This process separates compounds into small molecules and larger macromolecules, like proteins and nucleic acids. Living organisms also contain inorganic constituents, including water and ions like sodium and phosphate. Primary metabolites are biomolecules with specific functions. They are essential for growth, development, and reproduction. Examples include amino acids, sugars, fatty acids, nucleotides, DNA, and RNA. Secondary metabolites have different roles. They don't directly support growth or reproduction. Instead, they provide benefits like coloration, defense, and medicinal uses. Examples include pigments, alkaloids, terpenoids, and essential oils. Some secondary metabolites are toxins, lectins, or drugs like vinblastine and curcumin. Others have industrial uses, such as rubber, gums, and cellulose. Biomolecules are divided into two groups based on size. Micromolecules have molecular weights under 1,000 daltons. Macromolecules have weights over 10,000 daltons. There are four main classes of biomacromolecules: proteins, nucleic acids, polysaccharides, and lipids. Lipids are an exception, as they are included in the macromolecular fraction due to their role in forming membrane vesicles. In a cell, water makes up 70 to 90% of the mass. Proteins account for 10 to 15%, nucleic acids for 5 to 7%, carbohydrates for 3%, lipids for 2%, and ions for 1%. Proteins are linear chains of amino acids connected by peptide bonds. They are heteropolymers containing multiple different types of monomers. Each amino acid has a central alpha carbon with four key parts: a hydrogen atom, a carboxyl group, an amino group, and an R group. Amino acids are classified in different ways, including by ionization properties, aromaticity, and dietary requirement. At physiological pH, amino acids exist as zwitterines, with both a positive and negative charge. Proteins have diverse functions, including forming ground substance, acting as enzymes, regulating hormones, and defending the immune system. Collagen and rubisco are the most abundant proteins in the animal kingdom and the biosphere, respectively. Polysaccharides are large carbohydrate molecules made of many smaller sugar units. They can grow incredibly large, with molecular weights ranging from thousands to millions of daltons. Polysaccharides serve as structural building blocks or store energy. Examples include cellulose, starch, and glycogen, all made from glucose units. Cellulose provides structural support in plant cell walls, while starch and glycogen store energy in plants and animals. Polysaccharides have different structures, with two ends, a reducing end and a non-reducing end. They also come in different shapes, such as helical structures in starch and branched architectures in glycogen. Nucleotides have three parts: a nitrogenous base, a pentose sugar, and a phosphate group. The sugar can be either ribose or deoxyribose. Nitrogenous bases are divided into two categories: purines and pyrimidines. Purines have a bicyclic structure, while pyrimidines are monocyclic. There are 5 key bases: adenine, guanine, cytosine, thymine, and uracil. DNA and RNA differ in their sugar and bases. DNA has deoxyribose and uses thymine, while RNA has ribose and uses uracil. A nucleoside is a base attached to a sugar, but it becomes a nucleotide when a phosphate group is added. Proteins have 4 structural levels: The primary structure is the sequence of amino acids in a polypeptide chain. This sequence runs from the N-terminal end to the C-terminal end and is unique to each protein. The secondary structure refers to regular conformations of the polypeptide backbone, stabilized by hydrogen bonds. The two main forms are the alpha helix and the beta-pleated sheet. The tertiary structure is the three-dimensional folding of a single polypeptide chain, stabilized by various interactions. This folding creates the functional active site in enzymes. The quaternary structure describes the arrangement of multiple folded polypeptide subunits, like in adult human hemoglobin, which has 4 subunits. Enzymes are biological catalysts, mostly proteins, that speed up chemical reactions. They are highly specific and work at remarkable speeds. Enzymes lower the energy barrier needed to convert reactants into products by binding substrates at a special pocket called the active site. This process creates a temporary enzyme-substrate complex. The substrate is transformed, products are released, and the enzyme is ready to work again. Enzymes can be regulated by cells and respond to temperature and pH changes. They are vital for many chemical reactions in the body. A metabolite is a substance your body makes or uses during metabolism. A heteropolymer is a larger molecule made from different smaller units. Amino acids connect through peptide bonds, which are covalent links. Some molecules, called zwitterines, have both positive and negative charges. Sugars link together via glycosidic bonds, connecting monosaccharide units. For chemical reactions to happen, reactants must overcome an energy barrier. Enzymes have an active site where substrate molecules fit and bind. When proteins lose their shape, it's called denaturation, losing their structure. Competitive inhibition occurs when a molecule competes with the substrate at the active site, and this is reversible. An apoenzyme is the protein part of an enzyme without its necessary cofactor. Living organisms are very diverse, but their chemical makeup and energy processing are similar. This shows that the basic machinery of life has been conserved throughout evolution. Biomolecules are organized in a hierarchy, from basic building blocks to complex structures. This organization allows biomolecules to do many jobs while sharing a common foundation. Enzymes are crucial, making chemical reactions happen fast and efficiently. Without enzymes, life as we know it wouldn't exist. They drive cellular metabolism, which powers everything cells do. This reveals a deep unity underlying life's diversity.